So I've hopefully showed you all of the different parts of an experiment. I've showed you what the factors are, factor levels, etc. The only thing we haven't looked at is the actual experimental design, which is what we will look at now. So I'm back to my lemons. I've got my 24 lemons from Hy-Vee and 12 from Baker's. And we talked about how I might want to block these. And remember, I would block them because I'm concerned that the 12 Baker's lemons that are a little bit smaller might affect my results. And so I just want to do the experiment for each block of lemons. And we're going to do one treatment to be control, meaning the lemons are just at room temperature. And then our treatment group is going to be that they're placed in an 80-degree environment. And we're going to look at how we would design this. So I would start my experiment saying I had 36 lemons, and I break those into my 2 blocks. Now typically we like to put, an equal number in each group, but the fact of the matter is I have 24 Hy-Vee lemons and I have 12 Baker's lemons. And again, I blocked these because I feel that my Baker's lemons, which were a little bit smaller, might have a different result than my Hy-Vee lemons, which were a little bit larger. So within each group, I want you to notice that I'm taking my 12, breaking them into 2 equal groups, and that would just be randomly. This would be an SRS. I should have included that as well. A lot of times you're going to see SRS written here. So I'm randomly selecting lemons for each group. I put 12 in the control group, which is my room temp group. And then I put 12 in the treatment group, which is the 80-degree group. And then notice I'm comparing the juice output of just the Hy-Vee lemons. And then I look and do the exact same thing, SRS, with my Baker's lemons. I'm putting randomly 6 in my control group, 6 in my treatment group, and then I'm comparing the juice output of just the Baker's lemons. So again, when we have a block design, we're not comparing all of the results, we're only comparing the results within each block. So let's say I have 18 hosta plants, and I want to see how much water and sunlight affects the growth of the hostas. So I plan on giving the plants either 0, 5, or 10 hours of sunlight per day, and either 0 or 1 inches of water per day. We're going to do a tree diagram, but my question is, do I have to block these? And the answer is no, I don't have to block these because I just have 18 hosta plants, and there's no difference that we're aware of between those 18 plants, so there's no reason to block them. How many treatment groups are there? So blocking involved, no. How many treatment groups are there? Well, I know that plants either have 0, 5, or 10 hours of sunlight, so that's 3 levels of that factor. And either 0 or 1 inches of water, so 2 levels of that factor. So 3 times 2 would be 6 total treatment groups, which means at the end of my experiment, I should have had 6 different treatment groups and then compare those results. Here's what that might look like. I want you to notice the difference between what we did in our block design and what we have now. Here I have 18 hostas. I'm doing an SRS, a random sample, to put 6 in group 1, 6 in group 2, 6 in group 3, and typically whenever you're designing an experiment like this, each factor is going to have sort of its own column. So I chose to do the hours of sunlight first. If you would have just broken this into 2 groups, 9 in each group, and did water first, and then broke it into smaller groups to how many hours of sunlight, that's exactly the same thing, because the 6 treatment levels are going to be whatever happens in the second column. So here I have 6 hostas with 0 hours of sunlight, and then 0 inches of water daily. I should have put 3 hostas here, 3 hostas here. And again, my next group, group 2, 3 hostas, 3 hostas. Group 3, out of 6, I'm randomly selecting 3 hostas and 3 hostas. And you can put SRS on all of these as well. But I want you to notice now at the end, I'm measuring and comparing plant growth for all 6 of the groups, whereas when I did a block design, I only compared within each block. So now that we've practiced one of each type of the experimental design, I would like for you to look at scenario 1 and scenario 2. One of them should be blocked, one not. I'm going to not tell you which one is which, but what I would like you to do is read over these questions, and then on the following slides we're going to go through not only what the design will be, meaning what the tree diagram will look like, but we're also going to answer those questions like what are the factors, how many treatment levels are there, etc., etc. So press pause, take a moment to read these over so you know what we're dealing with, and then move on to the next slide. So the first question, and again, at this point, if you're like, hey, I want to try this on my own, this is the point where you press pause, do all of this, all of the questions, create the tree diagram, etc., and then press play. Otherwise, you're with me. We're going to start going through this. So we're going to look at a new drug called Bone Builder, and we're hopeful, or researchers are hopeful, that it will help children's bones heal after a break or a fracture. They're concerned it might work differently on bone breaks than on bone fractures. It will be used in conjunction with a traditional cast. And to test the impact of Bone Builder on healing, they recruited 18 children with bone breaks and 30 children with bone fractures. Design an appropriate experiment. So first, before we design our experiment, we have to answer these questions. Who are the subjects? In this case, it should be those 18 children with breaks and 30 children with fractures. The factor or factors and the number of levels for each. So what are the factors? Did you get Bone Builder or did you not get Bone Builder? That's it. So there's one factor, 2 levels, which is not Bone Builder, yes, Bone Builder. It doesn't give us a dosage. C, the number of treatments, because there's only one factor and 2 levels, the number of treatments is just 2. You either get Bone Builder or you don't. Is this experiment blind or double blind? Honestly, we're not given enough information to know whether or not it is. So it could be that some of the children were given Bone Builder and the others were just not. It could be that some were given a placebo and others were not. So if they were given a placebo, it would be single blind. If the people who are measuring the healing rate of the bones don't know who's in which group, then it would be single blind. If both groups don't know, then it's double blind. And then E, the response variable, that is, what is it that we are recording at the end of this? We're looking at whether or not Bone Builder helps bones heal, so we are looking at healing time. So I'm going to compare healing time for people who didn't get Bone Builder to people who did. Now to design an experiment, hopefully when you're looking at this, you see that we should be doing a block design. Why do I have to block? Because I have 18 children with breaks and I have 30 children with fractures, and they are worried that it's going to work differently on breaks than fractures. So what I want to do is I want to compare healing rate of breaks with Bone Builder and breaks without, and then fractures with and fractures without. So here's what my design would look like. I have 48 children total. I have a block for breaks. I have a block for fractures. I'm going to randomly assign groups, children into groups. The control group, which is no Bone Builder, and the Bone Builder group. And then at the end, I'm comparing bone condition with breaks, and I'm comparing bone condition with fractures. And my second question, an article in a local newspaper reported that dogs kept as pets tend to be overweight. There should be an S there. Vets say that diet and exercise will help those chubby dogs get into shape. The vets proposed 2 different diets, Diet A and Diet B, and 2 different exercise programs, Plan 1 and Plan 2. 60 single dog owners, which means there are just 60 dogs total, volunteer to take part in this experiment. Descriptions of each below. So Diet A, owners control portions of food and treats, and Diet B, Food is a mixture of fresh veggies with dog food and substitute treats with carrots. And then plan 1, 3 30-minute walks per week. Plan 2, 20-minute walks daily. So we're trying to see what is the best method for chubby dogs to lose weight. So who are the subjects? The subjects are actually the dogs because that's who we are looking at. We don't care about the owners. We're going to be looking at the dogs. What are the factor or factors and numbers for each? So there's 2 factors, diet and exercise, and each happens to have 2 levels, which means there are 4 total treatments. And again, how do we figure that out? 2 times 2. Now here I've listed what those treatments are, A1, A2, B1, B2, but it's not necessary to do that. Is this blind or double blind? It's not given, but it's at best single blind because obviously the people who are walking the dogs know who is in which group because they're feeding the dogs or taking them for walks, etc. But you could make a case that, well, we're not really looking at the people anyway, and the dogs don't know the difference. But we're not going to get into that whole discussion. Response variable, weight loss, because obviously that's what we're looking at. Is this going to help dogs lose weight? How would I design the experiment? There's no blocking here. I just have 60 dogs, so I want to randomly select dogs into each group. I'm going to have group A and group B, diet A and diet B. And then within each of those, I'm going to have 15 dogs for each plan. But again, notice the second column is my 4 treatment groups. So again, if I would have put plan 1, plan 2 here, and then I put the groups here, would I get the exact same thing? Yes, I would. Groups A, B. I would get the exact same thing because at the end I'm measuring the 4 groups, and the 4 groups are diet A, group 1, diet A, group 2, B1, B2, etc.